So that's me, Roger Meacock. Natural Healing Solutions has been my name of my practice since 2000. So today I'm going to talk about PCR because it's important, so the mod RNA potential for veterinary use, Bovaer, and a little bit on climate as well. So there's a lot to get through. It could do hours on each of these, so we're going to rush through as quickly as possible. So what are PCR tests? PCR stands for polymerase chain reaction, a test developed by Kary Mullis for which he received a Nobel Prize. It was developed as a clean laboratory test to detect a known sequence amongst other known sequences of RNA or DNA following gene therapy manipulation or to detect a known mutation. And I've put the bold in because it's really important. There's a subtle, really important aspect to this definition, which is that it is detecting known sequences amongst other known sequences. That's really crucial. And I didn't get that straight away. So it identifies a selective sequence, which may only be present in otherwise unmeasurable small amounts and doubles the amount present at each cycle of the PCR test. So the number of cycles used in generating a PCR result is important to know. So different primers are chosen according to the sequence of RNA DNA that is to be detected and amplified. So this is a little video that tells you how it is.
PCR is a powerful technique that allows us to make billions of copies of specific pieces of DNA. The first step in PCR is to open up the two strands of DNA to expose the genetic information encoded by the nitrogen bases, A, C, T, and G. In the next step, custom-made DNA molecules help us find the beginning and end of the DNA piece of interest among the billions of letters in the genome. These genetic bookmarks are called primers. The third and final step is to fill in the genetic information between the primers. An enzyme called DNA polymerase adds the building blocks of DNA called nucleotides to create an identical DNA strand. DNA polymerase works in our own cells to replicate our own DNA each time a cell divides. The first step in PCR is known as denaturation and is enabled by high heat. The second step is called annealing, and different primers have different and very specific annealing temperatures, typically between 50 and 60 degrees Celsius. The third step is called extension. DNA polymerase used in PCR works best at 72 degrees Celsius. In these three steps, we've taken our original DNA molecule and obtained an identical copy of a very specific fragment. If we repeat these three steps one more time, we'll go from 2 copies to 4. Repeat it again and we'll obtain 8 copies. And again, and we'll get 16, and 32, and 64, 128, well, you get the idea. DNA amplification by PCR is an exponential process. After 30 copy cycles, we will have 2 to the 30, or 1 billion identical DNA copies.
There we go. So that's PCR. We can pass an exam on it now. So the next slide is Kary Mullis, who's the guy who invented PCR, and he's saying what we can and what we can't do with it. In the video, he's talking a little bit about it with regard to HIV, because that was when he was alive, when he was talking about it. But obviously, it corresponds to whether it's bird flu or any other pathogen that they might be checking for.
I don't think you can misuse PCR. No, the results, the interpretation of it. See, if you can say, if they could find this virus in you at all, and with PCR, if you do it well, you can find almost anything in anybody. It starts making you believe in the sort of Buddhist notion that everything is contained in everything else, right? I mean, because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body, okay? So that could be thought of as a misuse of it, just to claim that it's meaningful. It is. No, there's very little of what they call HIV, and what's been brought out here by Phil Pott and Isai already. The measurement for it is not exact at all. It's not as good as our measurement for things like apples. An apple is an apple. You know, you can get something that's kind of like, if you got enough things that look kind of like an apple and you stick them all together, you might think it is an apple. But and HFV is like that. Those tests are all based on things that are invisible, and the results are inferred in a sense. PCR is separate from that. It's just a process that's used to make a whole lot of something out of something. That's what it is. But it's not. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything.
There we go. That's the key line, isn't it? It doesn't tell you that you're sick or that what you've got is going to be infectious. So what is PCR testing for? So as the video explains, only RNA and DNA with the correct sequence of nucleotides, which are just A, C, G, and T, that's all, will bind the chosen primer. So you get short primers, which are 18 to 20 nucleotides long, which provide faster binding but may have lower specificity, so they won't more things will cause a positive reaction, leading to false positives. Or you can have longer primers, where you've got 25 to 30 nucleotides, but they offer higher specificity, but you can get then folding of the primer or the DNA section that it's trying to check, which can obviously cause misreadings as well. So then you get false results from that as well. So if there's been a mutation within the primer sequence, then it won't be recognized. Or conversely, a mutation within a sequence that has actually then created what you're then looking for will also then be detected when it otherwise wouldn't be. And when we're talking about things like bird flu, which are frequently mutating, it sort of makes the PCR all that less reliable. So, okay, which of the following viruses are HPA, which is highly pathogenic AI, avian influenza, H5N1? Has anybody got any views?
No. They all look like man here.
The man here got it right. Answer: any, all, or none of them. If the section of the sequence of those recognised by PCR, and this is really crucial, they will, if they come up with a positive, they will say that's what it is. But it could be any one of anything else. But because it's come back as a positive on the PCR, it will be identified as what they're looking for. So what does a PCR positive actually mean? It means that the fragment of DNA that they've been looking for has been found that matches the primer. That's it. PCR cannot tell if the original RNA DNA came from a viable virus or how many of them there were. It cannot tell if the virus was capable of infecting the host and dividing. It cannot tell if there was a pathogen or a contaminant or part of the microbiome, or if it was mutated. It cannot tell if the immune system had already dealt with it and you just picked up a fragment of what the immune system had already dealt with. Without knowing the CT number, so the number of cycles that doubling process does, it is impossible to know if the original sample was capable of being infectious. For COVID, it was above 24 cycles was considered that the initial sample was therefore incapable of causing infection. But for these tests that they're doing, they're frequently cycling over 30 and even over 40 cycles. So once you've got to 30, you've got 1 billion times more than when you restarted with. And if that's doubling each time, I mean, you can work out how many times you've got by the time you've done it 40 times. It's an inconceivable number. So an analogy of this is if I was to take a swab from my sofa and look for my dog, there might only be a hair there, but they're telling you when you get back a positive, not only is the hair there, but the dog is too. That is the system where we've got to now with what's happening with PCR. So what is the risk of a false positive? Where the disease prevalence is low, then the chance of a positive result being false positive is very high. And I don't just mean like 50, 60%, it's 90-something% chance that it's a false positive. This is especially true when looking for HPAI, high pathogen avian influenza, in species other than birds and or in the environment, which then provides false epidemiological results as well. So where there's a lockdown, an exclusion zone, or a cull policy is in place, a false positive, as you know, has serious implications. If there is another cause of similar symptoms that can be affecting a population, then a false positive PCR means they won't go on to look for what has actually caused it. So it could be spraying, it could be vitamin E deficiency, which causes the similar symptoms, or you could even get some other pathogen as well. And it can, in an enclosed flock contracting HPAI, if there's no contact with the outside world, how has it happened? Common sense has got to come in here somewhere. If the police were looking at a crime, if there's no way for a criminal to get through the door, even if they can find a slither of that person's DNA in the room, and there was no way that they were ever going to be there because they've got an alibi as well, then they can't be convicted and they wouldn't get a result if they tried to go to court on that person. But that's not what's happening with the PCR test in the farming situation. They're not looking at the common senses. Even if we got a false positive here, how did the pathogen get in the first place? We've got to have some common sense. So how do we safeguard against false positives? False result figures should be established for all and any test in use, and they don't know them. PCR should not be used in isolation in a clinical situation. There was, going back to that picture of the viruses, and I said which ones are which. A good example of this that actually occurred was the president of one of the African nations. When COVID was being looked for, he sent off samples from goat, papaya, various other things, and they all came back positive. So that tells you how PCR is being interpreted. As Carrie Mann says, it's not that it's being used, it's being misinterpreted to insinuate that there was an infection there which couldn't possibly have been there. And this is what we've got to worry about and warn against for use on the farms. So PCR should not be used in isolation in a clinical situation from a swab with a highly mixed microbiome source. For those who haven't heard much about the microbiome, it's all the bacteria, all the viruses, all the fungi and everything, all the microbes that live on us. And roughly 10% of me standing here is human cells. 90% is microbiome. So when you're taking a swab of that microbiome, which has got sequences which they'll have no idea what's present in those other microbes, the chances of getting a PCR positive, they've got no idea whether it came from what they're looking for or anything else. And there's a high probability it came from some other source. And so this is where we've got this problem. So the CT number must be less than 23 as well, and certainly not the 30 to 40 as is commonly used. It should only be used where there are clinical symptoms. You can't go in where there's no clinical symptoms and screen for it, because it doesn't work. They did something similar in the States where they're looking at cattle, and they found it in an eye swab. If put it blunt, if a bird crapped in the feed trough and a bit of dust got in the cow's eyes, it put its head in the trough to eat. Of course, there's going to be something positive, possibly detectable there, but it's not infectious. It hasn't caused disease, but it's being interpreted as such because PCR told us it was. So it must be backed up with an antibody ELISA test or some sort of isolation identification. But even then, ELISA tests aren't without their false positives either. And as we know with TB, you get false positives with those too. And what they don't tell you is actually it's the cattle that are late in gestation, so they're getting close to actually calving. Those are the ones which are more likely to have a false positive. And the number of farmers where they're getting a positive on their farm, a lot of them are late in pregnancy, then it's quite highly probable that they are false positives. So as farmers, they should be asking what tests are going to be carried out and to make sure that you aren't exposing your animals to unreliable tests and unnecessary consequences, because it won't just affect you, it'll affect your neighbours as well, because there'll be exclusion zones put around too. But this needs legal advice as to where you stand legally. As far as I'm concerned, you've got an obligation for your animals not to expose them to a test which could be a high incidence of a false positive. But obviously, we need some legal advice, good legal advice on this as to where you stand on that, because if you can stand there knowing that you're backed up by law, then you're in a much stronger situation to go, no, you can't come on my land, because this test isn't reliable and we have evidence to show that it's not reliable, and therefore I'm not going to expose my animals to it. So I did some correspondence on this because I was pretty shocked that the PCR was being used. As it turns out, it's been used for a long time in veterinary medicine, but because I don't do conventional vet work, I wasn't using it and I wasn't aware that it was being used, but it is. So I asked the VMD, who the Veterinary Medicines Directive, why they'd approved PCR testing for clinical diagnosis when they weren't considered suitable by Carrie Mullis. So they wrote back to me and said, there is no authorisation process in place for tests. Nobody official or independent has verified their efficacy or established the figures for false positives or false negatives in different scenarios. And that will alter depending on the disease prevalence, obviously. But basically, they said to me, well, why don't you ask the test manufacturer? Oh, well, they're going to tell me the honest answer, aren't they? So whilst it carried on, the VMD forwarded my email on to the APHA, DEFRA, and they said, they came back to me and said, well, their staff know what they're doing. And it's like, well, I don't care about the nuts and bolts of doing the test. I know you can do that. But the replies didn't reassure me at all that they understood the interpretation properly, such as the risk of false results. Because my email, as you can imagine, was basically saying, what's the risk of false positives here? They did not address that false positives issue at all. If they'd known what it was, you could bet the bottom dollar they would have come back and said something to me about it. And all this correspondence, if people want to read the emails, they're all on my website, and I'll give you that at the end. So in my opinion, the PCR is being used inappropriately and interpreted inappropriately too. We risk a situation if we're not careful, as occurred with foot and mouth in 2001, where thousands of healthy cattle and sheep were culled for no good reason. What is the end game? I've actually written a letter to the Veterinary Times, which I'm hoping will be published, basically saying, what is an achievable end game here? Because we're not addressing the reservoir in the wild birds, and I'm not advocating a mass cull of those either. But we've got to work out what is a reasonable and achievable end goal where we actually have still got farming and we haven't decimated our national flock without having the ability to actually control what we're trying to control.